Welcome to this gorgeous space for the kickoff of Symposium 2022. My name is Sarah Witt. I serve as EOU's Chief Academic Officer and Provost. And it's a pleasure to welcome you this morning to the symposium after a brief hiatus in person during the pandemic. The Spring Symposium was an event spearheaded by Dr. Marie Balaban in the late 1990s. And its purpose is to showcase the achievements of our students and their demonstration of excellence in a course of study through the guidance and mentorship of their faculty. This is a time to celebrate the many ways our students achieve and will continue to achieve in contributions to society through experiential learning. Learning by doing and learning in the service of making a difference in real-world contexts and in service to the arts, sciences, and professions that express the possibilities of our shared human experience is the noble endeavor of higher education. I welcome you to enjoy the variety of formats the symposium offers. And I would like to introduce you to the architects of this year's symposium, library faculty, Sarah Ralston and Sally Milkey.
Thank you, Provost Witt. Spring Symposium is the culmination of months and sometimes years of work by EOU students and their faculty sponsors. We'd like to thank the faculty for involving students in hands-on research and experiential learning, and for the time they've spent ensuring the academic excellence that you'll see on display today. We would also like to thank the Faculty Advisory Committee, our administrative support team, Chris Martins, Leanne Slippy, and Kate Gepler, Devon Danaski and AV, Trevor Anger, the Custodial Services Coordinator, and Alan Arnson in the theater, for all the behind-the-scenes work that makes an event like this happen. Following this morning's keynote presentation, please check the symposium program for presentation times for posters, choral conducting performances, open art studio demonstrations, panel discussions, and talk presentations, which can be attended in person or on Zoom. Additionally, Oregon Teacher Pathway High School students will be presenting their research in the atrium of Badgley Hall. Here to introduce the keynote presenters is Professor of Chemistry, Dr. Annika Venado.
Thank you, Sarah, and good morning. It is my pleasure to introduce Brady Lehman and Lance Hatch as the 2022 Spring Symposium keynote speakers. Brady and Lance encompass all the characteristics of successful scholars. Not only they are smart and inquisitive, but they have demonstrated an amazing amount of commitment and resilience in their research pursuits. Brady Lehman will be graduating this June with a double major in chemistry, biochemistry, and molecular biology. Originally from Gresham, Oregon, he is a second-generation mountaineer. He joined my research group in February of 2019 as a freshman and has played a key role in the development of the project that he and Lance will be presenting. He has demonstrated an impressive independence, continuing to make progress on our project while I was on sabbatical leave last year in Colorado, even in spite of the restriction posed by the pandemic. His determination has paid off as we published just a couple of months ago our research in a peer-reviewed journal. That's a great accomplishment for an undergraduate student. But in addition to being a very accomplished scholar, Brady also has played so many other roles in our department. He is the president of the chemistry club. He is every possible class, I would say, organic chemistry, physical chemistry, physics, and its goal this past year was to inspire freshmen and sophomores to join chemistry, biochemistry research teams And early this year, he organized a research recruitment night that resulted in many new students becoming more involved in research. And nothing to brag about, but he was accepted at least in 8 different graduate programs. Small schools like Notre Dame, the University of North Carolina, Chapel Hills, University of Utah, just to mention a few. These are some of the most prestigious programs across the nation in analytical chemistry. This fall, he will be joining a Purdue University chemistry PhD program as a Ross Scholar with hopes to become involved in projects related to electrochemistry pertaining to biological systems, proteomics, lipidomics, and quantum biosensing. Lance is a junior in the chemistry biochemistry program, on track to graduate next year with a degree in chemistry and a minor in computer science. Originally from Walla Walla, he transferred to EOU in the fall of 2020, and soon after he joined my research group and started working on the development of the microfluidic biosensor that you will hear about today, making great progress. Lance is currently the chemistry club vice president, and I'm sure next year we will put him to good work. Lance is also planning to attend a graduate school after graduation with hopes to pursue a graduate degree in quantum science and engineering with an emphasis in quantum computing and artificial intelligence. I'm very, very proud of the work that Lance and Brady have done this year, and I just look forward to the presentation. So please welcome Brady and Lance as the keynote speakers.
Thank you, Anna, for that wonderful introduction. As Anna said, my name is Brady Lehman. I'm the senior group member in the Cavanado group. I joined in February of 2019, and then I started being the team leader in the biosensing project from the spring of 2020 to the winter of 2022. With me is...
Hi, I'm Lance Hatch. I am now the group leader of the biosensing team. My team member is Olivia Arvey. In addition to me, there's also Crystal Atwood, who's the head of paper microfluidics, with her team members being Zach Burns, Caitlin Cabelos, and Savannah Potter.
This is an extremely interdisciplinary project, and we'd also like to recognize the project's co-advisor, Dr. John Reinhardt of the EOU Biology Department. He's our geneticist here. So we're going to start off extremely general. What is the scientific process of science? So we start with asking a problem or identifying the problem that we want to answer.
We then go on to review lots of literature.
We plan and conduct experiments in phases.
And then continuously analyze results.
This is actually a cyclical process because the analyses that we get often lead to more problems and questions. Thus, we repeat this process until our hair turns gray. So, how does this look like in the modern era? Traditionally, there had been 5 disciplines of chemistry. However, now there's a breakdown and merging between them.
And with this breakdown, we end up with many subdisciplines, such as the one we're interested in, being nanotechnology.
This is indicative of the overall movement across STEM, with the breakdown and merging into
interdisciplinary and diverse teams working to solve increasingly complex problems.
And so this brings us to the first part of the scientific cycle, asking our analytical question.
And our question was, can we develop a biosensor to help identify bacterial kidney disease in salmon? So, living in the Pacific Northwest, I think everybody is aware of what salmon are. And I think the Columbia River Intertribal Fish Commission said it best when they said salmon are an intrinsic part of our spiritual and cultural identity. Now, dams play a very important part in waterways. However, they do have the drawback of hurting salmon spawning grounds. This is bad because salmon are what's considered a keystone species. They play a very important role in the ecosystem by both feeding predators and balancing the overall food chain. Thankfully, there are hatcheries, such as the one shown on the right, which is the Looking Glass in Elgin, which work to bolster repopulation efforts. However, hatcheries are not without their own problems. The main one plaguing both wild and hatchery salmon is bacterial kidney disease. It is a significant threat to the overall sustainable population of healthy salmon worldwide. It can be transmitted both vertically and horizontally, with the main causative agent being Renibacterium salmoninarum, or RS for short. In the top right, you can see a picture of how kidney bacterial disease externally affects salmon. And below that is a picture of kidney tissue that has been affected by RS. So here's a little overview of the literature we'll be diving into deeper. One thing I would like to point out is MSA, or major soluble antigen, as this plays an important role in upcoming slides. So, MSA is often the target of many primary testing methods. With screening, we have the direct fluorescent antibody testing, or DFAT for short, as well as the enzyme-linked immunosorbent assay, or ELISA, which is the most widely used. For confirmatory methods, we have a cell culture. However, those take far too long, clocking in at around 21 days. And there's also PCR, which is the primary confirmatory method. Tests are often conducted on ovarian fluid and kidney tissue.
So what Lance just mentioned were the current testing methods, and they have two main problems. A, they're time intensive and take manual analysis, and B, they cost quite a bit, so they're heavy on capital. For example, the multi-flow instrument used in the ODFW lab is used on ELISA, so this is an expensive piece of instrumentation. So overall, there's this movement towards lab-on-a-chip based technology. And in a traditional lab, you use liters of reagents, it's time intensive, and you can see that flow chart up above. However, with lab-on-a-chip technology, you can use 1/1,000,000th of a liter, a microliter, and have instant analysis. And so this microfluidics point-of-care testing is really what we're going forward. For example, I think we've all used one of these before. It's a COVID-19 test, antigen test. So this is an example of a microfluidic and point-of-care based test. It's a paper-based test. So there's a need across medicine, industry, environmental analysis, and other fields such as hatcheries for rapid, accurate, and cost-effective analysis. And when we view these different type of biosensors, there's many different types. But what we're going to focus on is DNA-based ones. Many of you, especially our high school scholars here, might know and understand about DNA and double-stranded DNA. So double-stranded DNA is at the heart of the central dogma of biology, which is DNA gets transcribed to mRNA, which gets translated to proteins. However, there's often a second type that's not really talked about, and that's single-stranded, artificially synthesized DNA. And you can think of this as similar to RNA in many instances, because it folds in upon itself, forming a secondary structure, as seen right here. However, it doesn't have the contamination problems that often plagues RNA. And so this is what we're going to term DNA aptamers, and they're a probe. And so the primary structure, or the linear forms, spontaneously folds in on itself, creating the secondary structure. And you can think of the secondary structure as a key that fits inside a protein lock. So here we can see the neuroprotein GRK2 having its aptamer fit in, C13 fit inside of it, and this was published in the Journal of Neuroscience. And this association, or the coming together of the two, is really, really tight, even tighter than classically used antibodies. Additionally, aptamers tend to be more cost-effective than antibodies too. And overall, this is governed by non-covalent bonding interactions. If you had an American teacher, you might know these as intermolecular forces. So how do we get to this really tight binding? And the answer is a system called systematic evolution of ligands by exponential amplification. But that's really long and complicated, so we call it selects for short. And there's many different types of selects. It's all based about what you're targeting, whether it be a whole cell, a protein, a metal, or other type of molecule.
So, with our question having been asked, and the literature having been reviewed, we were able to move on to phase 1, which was MSA characterization. So first, the MSA protein was sequenced. The subconstructs were then cloned and expressed using E. coli. From there, we used computation techniques to determine which part of the protein is most likely to bind to the ssDNA. In the figure on the right, you can see MSA and its subconstructs. The immunoglobulin plexin transcription factor domain, or IPT domain for short, is outlined by the box. Now, R2C had the longest IPT/length ratio. What this means is that it's the most likely to bind to the DNA, and thus it was chosen as our target. For phase 2 of experimentation, we reviewed ghost selects. Studies have shown that ssDNA binds tightly with Go. Therefore, graphene oxide, or Go for short, is an excellent tool to help enrich pools of DNA. On the right, you can see the structure graphite oxide, which is extremely close to that of graphene oxide.
And so this brings us back to the process of selects. Like Lance just mentioned, we're utilizing graphene oxide in this process. So the first step is ordering a library from a company. This library consists of an 80 base pair long aptamer. 40 base pairs of this in the centralized random region constitutes 10 to the 14th different combinations. And what this allows us to do is select for the specific ones that bind. Additionally, the primers on the end allow us to conduct PCR. And so the first step is to incubate this library with the MSA. Specifically, we're using that R2C subconstruct with the glutathione S-transferase tag. So it's a fusion protein, two subconstructs of proteins smashed together. And then we put in the graphene oxide because some of the aptamers don't bind to that MSA very well. And so we remove that graphene oxide ssDNA aptamer globule, if you'll call it that, via centrifugation, and then we disassociate the aptamer off the MSA. So now we have just the pool that bound to the target. Then we're going to undergo PCR. PCR is used to amplify, so make many copies of the DNA. However, it has two main problems. First is slippage, which is a mutation. You can see in lane 12s and 14, there's going to be smearing above the bands. However, lane 10 does not have that. Additionally, it has a very bright intensity, and that's exactly what we want to see. So we optimized for 10 PCR cycles. Additionally, we used a molecule called biotin, which has a super strong interaction with another molecule called streptavidin. So the PCR produces double-stranded DNA. We use a streptavidin-based column to bind the new DNA strands that have the biotin attached to it in the column, so we can use salt dilution and ethanol precipitation to get back to our next pool. We go through this process again and again, and we actually ended up with pool 9. We plotted the percent recovery through pool 7 for you all to see that as we increase in pool numbers through pool 5, we have an increased percent recovery. That means with each consecutive pool, we decrease the amount of aptamers we're removing from the pool. And what this shows also is that some of the aptamers were actually targeting the GST tag. We did a counter selects against the GST, and we saw a reduction in percent recovery, which is exactly what we expected to see. Pools 7 through 9 then returned back to that 80 to 90% recovery. So we removed the aptamers that were targeting the GST.
And with this, we were able to begin phase 3 of experimentation, which is the sequencing and computation of secondary structures. So, up in the graphic, in the top left of it, you can see pool 9, which Brady was just mentioning. We took pool 9 and we ran it for the 8 to 10 PCR cycles. We then took that fresh product and ligated or glued it into a topo-TA plasmid. That product was then introduced to E. coli, which was inoculated onto an ampicillin plate. From there, we selected for the white colonies and put those into terrific broth to grow. After it was done growing, we used a QIA prep spin mini kit to remove the plasmid from the broth. We then used that in conjunction with an ECO R1 restriction digest to confirm the insertion. And then from there, we sequenced to identify the secondary structure of the ssDNA aptamer.
And we'd also like to announce a very exciting news from the biology department. We just got 2 new sequencers this year from Thermo Fisher, one of them being a next generation solid state sequencer. This technology is brand new. It was just developed in 2018, as well as a more standard DNA capillary array sequencer. And we even got a brand new automatic prep station. We also like to pay respects to the instruments that we worked on, the Beckman Coulter. This served us and many other students in their research endeavors. So moving on, this is our primary sequence, or the sequence of DNA. I outlined and highlighted different regions of similarity among all 6 DNA strands. And so what's really interesting is that these primary or linear regions of similarity also correspond to regions of similarity based on the secondary structures. So we talked about the binding and folding before. This is exactly what a software called mFold is predicting. So we utilize computational methods to predict the secondary structures as well as the associated free energies. We want these aptamers to remain in this lower energy form, so we want the lowest free energy, and aptamer D ended up being the lowest of them all, so therefore it's our probe going forward with moving forward towards the biosensor. I also discussed how the aptamers have a really, really tight binding affinity in the introduction, but how do we tell that? We're going to utilize a procedure called immuno-qPCR binding affinity assay. This is a relatively new technique, but it can tell us how tightly the aptamer D binds to the R2C. So we're going to use the Roche light cycler located in Badgley Hall. And the first thing that we're going to do, that Lance is going to show you, is create a calibration based off of the threshold of the cycler to the logarithm of the aptamer concentration.
So here's that calibration curve Brady was just talking about. On the y-axis, you can see the cycle threshold, and on the x-axis is the log of the aptamer concentration. If you're familiar with graphs, you may notice that there is a negative slope. This is exactly what we expect to see, because with the increase in aptamer concentration, we want to see a decrease in cycle threshold. In addition to these great results, we also got a great correlation value of 0.9937. In addition to this, we also did the determination of the dissociation constant. On the left, there are a few values, but if there should be any takeaway from the slide, it's the constant listed on the graph, which is a KD of 3.0 plus or minus 0.6. What this means is that for every 3 unbound molecules to the DNA aptamer, we had over 1 billion molecules that did bind, which are great results. We also had a correlation coefficient for this of 0.99.
We'd also like to take a second to stop and recognize what Anna just said. This is the results where we published this past February or January in Molecules. We'll have a QR code on our poster out in the LOSO foyer, so you can scan that. This is an open access article and you can read the full text.
And with this, we can move into the 5th and final phase of experimentation, which is biosensor fabrication and electrochemistry. Now, with words throughout this presentation, such as nanotechnology and biosensor, you may think that there would be a complicated process to make these. However, that is far from the truth. We use a mix of carbon, ink, and graphene oxide to screen print the electrodes onto a transparency similar to that of a t-shirt. Brady is actually holding one up right now, but if you can't see that, we also inserted a picture into the slide. On the left side of the biosensor is the reference electrode, which has been painted with silver chloride. In the middle is the working electrode, and on the right with the overhang is the counter electrode. So now we can finally get into everybody's favorite subject, electrochemical theory. We work with what is known as ferricyanide, which can be seen as FeCN6 with a 3- charge. Now, when we put this onto the electrode, as shown in figure 26, the FeCN6 3- moves in and makes contact with the working electrode. Now, when it moves away, it's still FeCN6, but now it's 4- because a reduction reaction has taken place. I can't stress how important it is that the ferricyanide make contact with the electrode for this reduction to take place. In addition to this, the redoxivity of the system is proportional to that of the current running through the electrode. So here we have a schematic of the electrode system. On the far left, you can see a bare electrode, where the ferricyanide is free to come in, make contact with the electrode, and the reduction can take place. In the middle, there's an aptamer-modified electrode. Now, this kind of gets in the way of the ferricyanide. However, it is still free to make contact, and that reduction can still take place. On the far right, you can see an electrode in the presence of MSA. MSA is a much bulkier molecule than ferricyanide, so it almost blocks it from being able to touch the electrode and that redox reaction from being able to take place. Now, as modifications are made to the electrode, the resistance goes up and the peak current goes down via Ohm's law. Speaking of Ohm's law, you can see it illustrated in the bottom of the slide as V equals IR, where voltage is equal to current times resistance. So at the same voltage, in the presence of MSA, there is more resistance than the aptamer-modified electrode. Thus, the current should decrease because the ferricyanide cannot make contact with the electrode, making it so the reduction cannot happen, thus rendering the system irreversible.
So I really want you to pay attention to that term Lance just used, irreversible system. This is what we're going to be capitalizing upon during our biosensor making. But before we can even quantify the MSA, we have to learn and see how our electrode and electrochemical system actually works. So we selected one of our fundamental experiments to show you all. So this technique is called electrochemical impedance spectroscopy. You can think of impedance as the AC version of resistance, which is DC. And so we want to see this impedance or resistance increase when we attach the aptamer. And what we can see in what's called a Nyquist plot is that the blue curve is going to be the bare electrode, while the red curve is the ssDNA aptamer modified electrode. And we see this area underneath the curve increase and the height of the curve increase. And so this area and height is proportional to the impedance or resistance of the system. So we see an increase in impedance as we attach the aptamer. And this is exactly what we want to see. So we're going to move on into our main electrochemical technique, which is known as cyclic voltammetry. Voltammetry is when you apply a voltage or an electrical force to the system while you're measuring current. So everybody remembers V equals IR. And then it's called cyclic because we start at one point of the voltage, so we start applying one force, but then we actually ramp it up, we stop, and then we ramp it back down. So we start and stop in the same place. We make a circle, hence the term cyclic. And what's shown here in this figure is that up above is a reversible system, and down below is an irreversible system. And there's two main points and items that I want to discuss with this. First being the peak height. Lance discussed how the peak height should decrease at the same voltage, and that's what we see here. So going from a reversible up above to an irreversible down below, at the same voltage, that current decreases. Additionally, we can also view something that's unique to the irreversible system. So first looking back up top at the reversible system, we can see the two peaks, the positive and negative peaks, are really squished together on the x-axis, right? They're pretty much right next to each other if you drew a line down to the x-axis. However, in the irreversible system, there's this stretch along the x-axis, and this is really important. Moreover, we refer to that as delta delta E, or the difference in the change of the location of the peaks along the E or voltage axis. And so we're going to be capitalizing upon both the peak height and the stretch. So theory is nice. I'm an experimentalist. I like to run experiments. So we're going to first look actually at figure 31. This is plotting the stretch of the system as when comparing the blue bars, which is going to be the bare electrode, with the orange bars, which is the ssDNA aptamer modified electrode. And we can see that increase in stretch showing the more irreversibility of the system. Furthermore, when we have the system in presence of 21.6 micromolar, so that's 1/millionth of a molar, which is a standard concentration that we use, we can see additionally a stretch going from 0.26 millivolts to 0.30 millivolts. So it is making that stretch again. And then we can look at the peak height. When it's in presence of MSA, there is a 20% decrease in peak height going from the bare electrode down to the MSA presence electrode. And this is exactly what we want to see. So telling there's a difference is nice, but quantitation is key. Can we calibrate it? Can we tell how much MSA is present? And the answer is preliminarily yes. So this is a cyclic voltammogram that we took. The red curve is going to be the ssDNA aptamer modified electrodes. The other three curves are going to be in the presence of MSA. As you can see, there's that stretch that we want to see additionally, showing the more irreversible system. Also, we can view the green curve as being 21.6 nanomolar, so nano being 1/billionth of a molar. And then the salmon color, pun intended, pink curve is going to be 100 nanomolar, and then the blue curve is going to be 216 nanomolar. So what we can view is this inverse relationship between the concentration of the MSA and the peak height, which is exactly what the theory told us, so that's great that we see that. And in fact, we can plot a preliminary calibration curve. And so this is over our first 10 electrodes. The first thing you might see is a large error bar, and some didn't work because the modification didn't take place. However, when we average over 10 different electrodes, we see a halfway decent R-squared value start to take form with a 0.9794, also 0.98 calibration curve. And so this is based off of that decreasing peak height, so we see that negative slope or the inverse relationship. And this takes us to where we are currently. And we have three main immediate objectives moving forward. First is to evaluate that stretch for its ability to also create a calibration curve. So we looked at the current through the system, but can we look at that change in the voltage where that peak is? Additionally, can we evaluate what's called a capacitance measurement? We mentioned other fundamental measurements we did. This is one of them, and we want to see what this non-faradic, we just discussed faradic here, current can tell us. What does the charging on the actual electrode tell us? Additionally, we want to increase the robustness of these electrodes. We want the fisheries to be able to do this technique, so therefore it must withstand these heavy environmental factors.
So in addition to the three immediate objectives Brady just mentioned, the team also has 4 long-term objectives. The first and primary one is to reduce the error found in that calibration curve that was just shown. The second is to determine the limit of detection for each different method. And the third and the fourth are fairly similar, and that for the third, we want to test the biosensor against an RS positive sample of water, and the fourth, we want to test the biosensor against a positive sample of kidney tissue. So with what time we have left, we'd really like to make some acknowledgements. The first of those goes to Anna Cavanato and John Reinhardt for advising us through this whole process. We simply couldn't have done any of this without either of them.
And you might have seen we covered a lot in this presentation. It takes a village to do research. Luckily, we have very enthusiastic undergraduate students here. I'd like to recognize all the students that participated and were team leaders even before me that worked on the the current ongoing electrochemistry, the previous qPCR binding affinity assay, as well as the selects process. For funding, we would like to recognize the Society for Analytical Chemists of Pittsburgh for their Analytical Undergraduate Research Program grant, as well as EOU.
For travel support, we'd like to thank the National Science Foundation Scholarship, the ACS Richland Local Section, the ACS, and EOU.
For helpful discussions, we'd like to thank Dr. Chuck Henry for hosting Anna on her sabbatical last year out at the CSU Department of Chemistry, as well as EOU's Dr. Colin Andrew from the Chemistry Biochemistry program, Dr. Joe Corsini from the EOU Department of Biology, both for helpful discussions, different parts of the project. And then also Sally Gee with ODFW, who hosted me on an internship last year in fish pathology located here in LeGrand, and Sarah Ralston for very helpful discussions about how this talk should look.
And here's that literature we mentioned at the beginning.
We're going to be opening up the floor for questions now. So that's one of those fundamental questions that we're looking at. Anna, I believe, did some experiments on that. What did you find? Do you remember?
I only ran some preliminary experiments when I was in Colorado, and it looked like if the electrodes were kept at a refrigeration temperature, without really any stabilization or anything, they would last about a week. And of course, that's not something that you could commercialize, so there needs to be some other way of stabilizing the surface of the electrode, which is commonly done because there are other electrochemical type of sensors. So it's something that we needed to further investigate.
Thank you. Yes, Dallas.
So you talked about the fact when you're making the electrodes and everything, it's essentially like printing off a t-shirt. It's similarly cheap, like printing off a t-shirt? Is it cheap to produce and screw it? Or?
It is pretty cheap to reduce. I think, what are we at now, about 8 cents a unit?
Yeah, and this is like contrasted to a platinum or gold-based electrode. Yep.
For future testing, when you address actual tissue values, are they going to need to go through the PCR process as well and find the aptamers?
Okay, good question. For the tissue level, it's going to take some extraction, so like using a solid phase extraction technique to just get it down to where it's a more simplistic matrix along with the R2C. So homogenization and probably solid phase extraction, more on the technical side. Who else has a question? But it can also be like about our time at EOU too. It doesn't have to be technical. I know there's lots of high schoolers here.
How did you guys decide to choose like chemistry? What made you take this as a study?
Well, for me, I frankly found every other subject not that fun. And also pretty good employment rates post-degree, so.
Lance also did mention that I was his roommate last year as well as this year, and I bullied him into it. I love solving problems, and so I did both the biochemistry and molecular biology paths, and the research has allowed me to help solve a real world problem. And so working with my hands, I mentioned that I like to do experiments. I like the theory, but I really like the experiments. It's really intriguing to me. Yeah.
Thanks for your talk. I'm curious about your future, like leaving for grad school, and how you're going to feel about leaving this big project behind, or are you leaving it behind? Will you continue to experiment with your colleagues here at EOU? And
So.
same for you, Lance, like when you move on, like, how does that process work?
So Lance is now the head of the team, so I have taken a step back. Most of my day in research is actually filled like I'm preparing a manuscript for a different project right now. So I do a lot of writing right now. I'm very sad to leave EOU. Anna has been a great mentor to me as well as the other chemistry and biology faculty. But I do want this to be an undergraduate-based research project. And so that's my reason for not really continuing on with it. Other professors have offered me spots on aptamer-based projects. I didn't end up choosing the school, but I got offered a project based on a biowarfare agent for the Defense Department out of school. So having this amazing solid foundation is really important. EOU is really a powerhouse among the chemistry research nationally for undergraduate research.
So what Brady didn't mention is that most likely he will become EOU's first chemistry graduate student, as we will be emailing him very often to ask questions. As far as I'm concerned now, under me I have a very competent team member, Olivia Harvey. Hopefully she'll be taking it over after me, so there's no worries whatsoever on my end.
Yeah, just to bounce off what Lance said, something unique about our program is we bring on freshmen, looking at you high school students. We build longevity. That way you're essentially independent by junior year. Lance started last year as a sophomore, but he's independent now and able to direct experiments even when Anna's not there. So that's something unique we do here, having 3 to 3.5 years of research experience. Other questions? Yeah.
So after you entered the research project, how much time did you feel that you had to explore other aspects of research? Like games and like sports and fun activities.
We're not really familiar with fun activities. But I mean, as far as for me, like Brady said, I was his roommate, so I was indoctrinated into research day one. So that immediately started taking my time anyway. I'm not sure how it was for him.
Yeah, but we do have time to like hang out. All of us in the chemistry department are extremely close. Some might call me a workaholic. So I find it's pretty easy. We also have chemistry club. We have that poster that I'll be at also that does extracurricular activities outside doing outreach and education. But I find a very good quote-unquote work-life balance between our primary academics, the research, the chem club, and just relaxing and having fun and being a normal college student. Other questions?
Dallas?
So would you say you're a party animal? Like,
Yeah, man, I mean, we get pretty crazy in the research lab. You know, sequencing DNA, and cloning E. coli, all that kind of stuff. Oh, yeah.
Yeah.
On the record, we have very good lab safety practice. Oh, yep.
How many hours do you guys do this?
I can recall nights where I was there with the sequencing project at 3 a.m. When you're dealing with biologicals, they dedicate your time. They dictate your time. You don't dictate your time. Probably thousands, quite frankly. But I love research. This is not work to me. This is something I love to do.
Yeah, let's just say you get very familiar with the centrifuges and micro droppers, that's for sure. Are there any other questions? Oh, Savannah.
So in terms of the electrodes, are you hoping to have this available at multiple hatcheries or specifically the one you mentioned in your project? And how feasible is it for the hatchery workers to do it on site versus having to have really advanced technology to help interpret the results?
Well, as far as hatcheries go, we're definitely not just going to limit it to looking glass and alginate. That would be a little odd if we just started denying people to use it. But I think for a while, to make it worker-friendly, instead of having to use a palm sense, which is thousands of dollars, we were looking into fluorescence a little bit just to make it more straightforward for the workers so they don't have to learn about physics and Ohm's law and electrochemical theory and whatnot. But I'd say that's still a little bit of ways out. There's definitely still work to be done.
The goal would probably be to partner with a private enterprise to make an instrument that can either give a positive or negative or I don't know answer. Sensor development is often, the base of it, what you just saw, is done at universities, but oftentimes there's industrial partnerships that take place moving forward to kind of wrap it up and deliver the final product. But we're still years off from that, quite frankly. We have to get that error bars down and make it more robust. Good question. Any other questions?
Caitlin?
So you mentioned that this was specifically for bacterial liver kidney disease in salmon. Do you see this type of method being used on other fish with other diseases or illnesses?
So maybe not fish, because fish are often not widely created biosensors, but like what I was mentioning, anthrax was being currently developed. There's actually an aptamer-based delivery system, so we talked about sensing. There's also other methodologies for like lipid bubble delivery systems. It was some type of eye disease that I saw. And there's lots of different human-type diseases, including like PKE, phenylketonuria. I did my seminar this past year on the aptamer-based sensors. So there's tons out there. Any other questions?
Yes.
What made you guys choose EOU?
Good question. I was looking at some very, very larger, big programs. EOU offered me the ability to jump right into research my freshman year, and so being a STEM-based student, I just felt this sense of community where all the students knew each other, and all the professors were on first-name basis. I have all my professors' phone numbers on speed dial and stuff if I need them. It's a very kind of nurturing place that everybody can succeed at.
Brady always has a scientific kind of process. I just pick names out of a hat pretty much, so I end up here. Yes.
There are often structural barriers to working in an interdisciplinary way. What lessons did you learn that you're going to be able to use in the future for approaching projects in an interdisciplinary manner?
Yeah, good question. I know I struggled initially with the sequencing. Something that happened was we were getting a lot of false negatives, and I think actually Dr. Corsini, I know he's out there somewhere, pointed out that you can, because it's so short, the protein can actually be functionable even if you have insertion. So just talking and being open, and that's a lot of the soft side of science, is being able to communicate well. And having those interdisciplinary discussions is really important, and we see that across. When I was visiting, 8 different grad schools this past year, I saw these interdisciplinary teams really starting to flourish.
I think in addition to that, part of it is being able to relinquish control of your experiment onto other departments. You spend hundreds of thousands of hours with these things, and you become very intimately familiar, and it becomes very personal. So being able to take a step back and let someone else give their input, or run an experiment, or give you data, I think is a really big part of it. Is that everybody? Maybe.
Yes. So you're going to be a senior coming in here to here. Excellent question. So if you're like biochemistry, molecular biology, chemistry, you're going to be taking general chemistry, first with Ron Kelly, then Colby Heidemann, and then Anna. You're also going to be taking general biology. Dr. John Reinhardt teaches the last 2 sequences, and then currently Karen teaches the first sequence of it. You'll also take physics sometime when you're here, whether that be a freshman, junior, or senior year. But yeah, and also having a really great writing foundation is super important. I know Dr. Marta's down here. She really stresses writing in her classes as well. So does Anna in her chemistry classes. I think I did a lot of college and high school, and actually the most important class I ever took was my writing 121, 122 class. Any other questions? Who here is in high school attending? Awesome. I love seeing the future rising stars of Eastern, especially with your research. Yes?
All right, so I got a question for you. A lot of incoming freshmen are probably not going to have all the same connections you guys do right now. So getting onto a research team can be a very intimidating process for them, because they're going to have to get acquainted with a lot of people and make sorts of connections. So how would you advise they go about making those connections, putting in their name with some people, so that they can actually get onto a research team and have all the same opportunities as y'all?
Well, thankfully this year, Brady established that research night where students are free to interact with staff and just kind of see what research is going on and able to talk to them about joining teams. So I think that was a very good step in the right direction. Outside of that, just send them a lot of emails and annoy them. I guess there's Chem Club too, but really just email them a lot.
Chem Club initially, also, all of our professors are really involved with the chemistry club. We're extremely active on campus, so that's more FaceTime with professors. It's very easy to get on research here compared to other schools. We're extremely eager to have new students on because we're just undergraduates. We don't have graduate students. There's always, you're here for 4 years, we have to keep this cycle going. And to have the students on for a long time is really crucial.
Maybe one more question?
Dallas?
So, like, you guys from Walla Walla, right? Correct. Did you ever feel like you were maybe behind in getting onto research stuff? Like, obviously, Brady was your roommate, so it did help that you got to, like, shove you into a corner until you're doing research. ever feel like behind or anything from having transferred in rather than being here all 4 years?
At no point did I ever worry about being behind. That's for sure. I mean, that could have been circumstantial, but like I said, we now have different meetings and programs in place to pair different students with researchers. And you know, it's not like an audition. Like these instructors, they want research students with them.
This chemistry recruitment night we've mentioned a couple times is going to be a formality from this point moving forward. So we make sure all the students have the same opportunity to get onto these prolific and important teams. I know we have, other than Lance, another student that transferred in, and then we also have multiple freshmen on our team as well. Excellent.
So during COVID, did you guys still do the lab?
Yeah, excellent question. One of the reasons, beyond Lance's obvious brilliance also, the reason why I chose him as a research partner is we were roommates. That limited our contact, and we often would work at very odd hours, like 9 to midnight, to minimize contact so we could still be productive. So we made sure that we were acting in a social distancing back then responsible manner. But we were still able to make quite a bit of progress, even when Anna was out at Colorado State last year. The one term we went online, it was more on literature review, moving more into the biosensor, so I was still able to be very, very productive, even online.